All right. We are back with some OXPHOS, our affectionate little name for oxidative phosphorylation. I've pre-drawn my membrane, so if you're wondering where we are, here's our mitochondria. And inside the mitochondria is this inner membrane. Okay, so there's the outer membrane, which is this one right here. Okay, there is the intermembrane space. Okay, inter meaning between, so the between membrane space, which is right here. There is the inner membrane. Okay, so that's the one on the inside. And then, of course, as we mentioned earlier, in the very center is what we call the matrix. Okay, and it's like a jelly-like fluid. The folds, so when you look at these folds, which increase surface area, that is called a cristae. So sometimes you'll hear me or other people kind of use cristae and intermembrane interchangeably. Technically, the cristae are the folds. And that's supposed to be an arrow pointing to those. But a lot of times I'll just refer to the intermembrane as the cristae. So this reaction, and any time you're going to have an electron transport chain where you're creating a gradient, there has to be a membrane involved. Okay, and so that's kind of a clue. So this is happening along this inner membrane, which is a phospholipid bilayer. Okay, just like the outer membrane, just like the cell membrane, just like the membrane that makes up the ER and the Golgi, all of those are phospholipid bilayers. I didn't put all my little tails over here because I'm going to be drawing some stuff in there. So I didn't want that to be a little confusing. Out here is the intermembrane space. Okay, and down here is the matrix. So we just did the citric acid cycle down here. Okay, so that kind of gives you a feel for where we're at. And what we've got going on here is we have this membrane, and embedded in this membrane are these proteins. And these proteins are, for the most part, proton pumps. Okay, they have different names, doesn't really matter. We don't need to know that much detail, okay? But they're basically proton pumps. And so what we also have going on, I'm just trying to set the stage, I'm setting the scene before we say action, is we have some hydrogen ions hanging around. Now, hydrogen, if you'll remember, is a very simple molecule. It has one proton and one electron, no neutrons, if it's the common hydrogen. And so what happens is when we strip away the electron, which makes it positive, then we're left with just a proton, okay? Because if it's just an electron and a proton, and we take away the electron, it's just a proton. So another thing that you could call these hydrogen ions is a proton. Okay, so when we call these proton pumps, they're going to be pumping hydrogens. Okay, that's another way of saying it. All right, so I think we've got the stage set, the scene is set. So now let's bring in our actors. The first one to come in is going to be NADH. And remember, NADH is carrying electrons. NADH is going to come in. I'm going to zoom in a little bit just to help with my focus. And it's going to drop off the electrons. Okay, and those electrons, let me put them in a different color. We'll do them in green. They're going to go into these proton pumps, these electron transport chain molecules. In doing this, this becomes oxidized. So this is the first time you've seen me write oxidized near NAD+. It's always been reduced because we've always been going this way. But now we're removing those electrons and it's being oxidized. It is then going to leave and go back to glycolysis and pyruvate oxidation and the citric acid cycle, all those things to pick up more electrons. The other actor that you know that's going to come in and do the same thing is FADH2. It's going to drop off its electrons, okay, which is going to oxidize it. So it's going to get oxidized back to FAD++. And it's going to drop off its electrons to the electron transport chain as well. All right, so what happens to these electrons? Well, they get passed down. So when this molecule right here, this blue circle, accepted these electrons, it became reduced. It is then going to pass those electrons to the next molecule. When it does that, it becomes oxidized and that molecule becomes reduced. This molecule also becomes reduced when it takes electrons from FADH2. Okay, it's going to pass its electrons to the next molecule. So when it does that, it is now being oxidized. This one is being reduced. It's going to pass its electrons to the next molecule. When it does that, it has been oxidized. This one that just received the electrons is being reduced. And this one says, you know what, electrons, you are just about out of energy, so you're done. And so it's going to give those electrons to oxygen, which is going to join up with some of these hydrogens to make water. Now, I know that's not balanced. It's okay. Don't stress out about it, okay? So oxygen is called the final electron acceptor because it is taking those electrons here. Now, if you heard me, I said that this one said, all right, electrons, you don't have much energy left, so you're done. What happens as it moves, as it passes these electrons down, as it does those redox reactions of, I receive that electron, I'm reduced, I give that electron, I'm now oxidized. I receive an electron, I'm reduced, I give an electron, I'm oxidized. I am oxidized. What happens then is that the molecules, the proton pumps, they say, okay, electrons, we will pass you down, we'll do you that favor if you'll give us some energy. So the electrons pay these molecules in energy. So as the electrons get passed down, they lose a little bit of energy at each step, giving that energy to these molecules, and they use the energy to do active transport. So they're going to pump these hydrogens from a high hydrogen concentration, from a low hydrogen ion concentration to a high hydrogen ion concentration. So when you pump low to high, let me zoom out a little, whoop, wrong way, zoom out a little bit. When you pump low to high, that's going against the concentration gradient, and it does require energy. And so that's what's happening here is these proton pumps are using the energy of the electrons to pump the hydrogens up. What that's going to do is that's going to increase this hydrogen ion concentration even more. So as if there weren't enough hydrogens up here, we're going to cram some more in. You could take the pH of this. This would be very acidic because it does have a lot of hydrogen ions. That's just a random side note, random fact there for you. And as I mentioned earlier, this does have to occur across the membrane because you've got to have that semi-permeable membrane to create this gradient of high concentration and low concentration. Well, meanwhile, while all these hydrogens are getting pumped up here, because this is happening again and again, more NADHs are coming in, more FADHs are coming in. As they come in, they drop off more electrons. That gets passed down. So you're constantly pumping hydrogens up, up, up. These hydrogens are starting to get a little cranky. They're like, whoa, 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 whoa. There's too much. We're crowded. I don't want any more. And they're looking for a door. Well, the only door to escape is this enzyme called ATP synthase. Okay? So they're going to fall back through this door. This is going to be passive transport because now they're going from high concentration to low. So it's going to be passive transport, facilitated diffusion because it is going through a molecule. And as it does that, it's going to cause this molecule to literally start to spin or turn. As it turns, it gives the energy needed to phosphorylate ADP into ATP. Okay? So this is called oxidative phosphorylation. Phosphorylation because we're adding a phosphate group to a molecule, ADP, and making ATP. Oxidative because it is powered by these redox reactions where these electrons are moving down. Those are a bunch of redox reactions that give the energy to pump the H's, which gives the concentration gradient to allow the H's to fall back through. Okay, so where before we talked about substrate level phosphorylation, an enzyme was doing it. Here it's called oxidative because oxidation is powering. It's giving the energy for this phosphorylation. All right, couple last things. Sorry, I have a tickle in my throat. This is broken down into two parts. This first part that kind of ends right here is the electron transport chain. Sorry, guys. Goodness gracious. The second part, all right, I think I got most of it out. I had to drink some more water. The second part is happening here. It is called chemiosmosis. Okay, osmosis, because we're moving molecule from high concentration to low. I know osmosis is usually water, but since hydrogens are a part of water, we call it chemiosmosis. And those two things together make up oxidative phosphorylation. So let me zoom out a little bit more. This is going to make, that's not real pretty, but this is going to make about 34-ish ATP in a perfect world. It's not always going to be perfect, but 30 to 34 ATP. We made 2 ATP in glycolysis and 2 in the Krebs cycle, or the citric acid cycle, so that's a total in a perfect world of 38 ATP. A couple of things about this. It does occur along the inner membrane. It is aerobic because of these oxygens right here that come in. They're atmospheric oxygens. And it does not do substrate level phosphorylation. It does oxidative phosphorylation. Okay. And so let's see. I can describe the process. I can list the reactants. So let's do the reactants real quick. And we don't have to worry about per glucose or anything at this point because we're getting all those NADHs. Reactants are NADH, NADH, FADH2, some oxygen, some ADP. I guess that's all. Okay. And then my products, let me keep with my color code so you'll know what's going on. My products are NAD+, FAD++, water, and a bunch of ATP. Okay, and again, we don't really worry about numbers here because it just depends on how many NAD pluses or NADHs come in and all that good stuff. So I can list the reactants, I can list the products, I can explain how the electron transport chain uses redox reactions to create a hydrogen ion or proton gradient. So we talked all about that. I can explain how chemiosmosis uses that hydrogen ion or proton gradient to produce ATP. We talked about that. And I can explain the role of ATP synthase, which is this enzyme right here, in oxidative phosphorylation. So that is all of the I can statements. I hope that helps. If you have questions or want me to go over it again, like I said, I always recommend drawing it out a couple of times. It helps get it in your brain. But if you have questions or want me to go over it again, just let me know. Thanks, guys.